Hello, my name is Ethan Colson. I'm a student at Augusta Technical College, and I'm actually taking a biology course there now. For one of our projects, we have to make a video explanation of cell division. So here we go. So drawing a cell, I couldn't get every organelle just because there's a lot of organelles, and I only drew a few to make the cell look full and then also to get the main points of cell division. So before interphase, the cell actually lives in normal conditions, right? It's just living its life. This is, at times it's actually called G0 phase, but it's not that important. But the cell is just living its life. Now, after interphase though, and during interphase, a lot goes on. So interphase actually has 3 parts and 2 checkpoints. So in interphase, the cell prepares for the mitotic phase by duplicating the internal organelles and also duplicates the DNA. The checkpoints confirm the cell is ready to move on in the division process, and also the S checkpoint actually confirms that the DNA was correctly duplicated, that the DNA is correct. And after interphase, we see prophase. And in prophase, the chromatids coil, and it can be seen under a light microscope. The nuclear envelope begins to break down, and the Golgi apparatus and ER move to the outside of the cell. Spindle fibers begin to emerge from the centrosomes, and the centrosomes move to opposite poles of the cell. After prophase, there's prometaphase, and in prometaphase, the chromosomes connect to the cytoskeleton, and the remnants of the nuclear envelope are gone. As you can see, there's no more that shade of purple. There's no more nuclear envelope, it disappears, and each chromatid attaches to spindle microtubules via kinetospores, which are a protein. And after prometaphase is metaphase. And in metaphase, each sister chromatid is attached to the meiotic spindle, and they're lined up on the metaphase plate. Now there's actually a checkpoint in metaphase. It's called the M checkpoint. And in the metaphase checkpoint, it confirms that every sister chromatid is correctly attached, because this phase is unreversible. If they're broken incorrectly, it could lead to some serious problems. And then on to anaphase. In anaphase, the sister chromatids are split and become chromosomes. The chromosomes are pulled to opposite sides of the cell, and the cell actually becomes elongated. And next is telophase. Now, telophase looks a little bit different for cells with cell walls and cells without cell walls, but we'll get to that next. So in telophase, all the events in the first 3 parts of the mitotic phase are undone. The chromosomes now move to opposite sides of the cell, and they start to uncoil, start to unravel, and the nuclear envelopes actually form. The chromosomes, yes, like I said, reach the opposite sides of the cell, and the spindle fibers become monomers for the new cytoskeleton. And there wasn't really a way for me to draw that, but all the spindle fibers become the new cytoskeleton. Now, cytokinesis, which is what is different for plant and animals, or ones without cell walls, ones with cell walls. We'll get to that now. So for cells without cell walls, actually, after anaphase, a contractile ring forms at the metaphase plate. This ring comes together to form what's called the cleavage furrow. The furrow deepens until it separates the cell. Imagine like they put a rubber band, and it tightens slowly until the cell splits apart. Now for cells with cell walls, we actually have to back up a bit more. So back all the way in interphase, when the cell's getting ready for division and collecting the necessary glucose and different proteins, the cell actually creates extra glucose and extra structural proteins to make sure it can handle the division. It actually puts the glucose and structural proteins in these vesicles. And in telophase, these vesicles travel down the meiotic spindle to form a cell plate, which becomes the cell wall. And the membranes from these vesicles actually become the new cell membrane within the cell wall. After that, our last topic is human somatic cells versus human gametes. And this sounds complicated, but it's really not. In a human somatic cell, which is a majority of our cells, right? Majority of our cells are going to be somatic, also aka diploid. And all that means is they have two sets of each chromosome. One from our father, one from our mother. But human gamete cells are also called sex cells, and they're used for reproduction. And they only have one set of each chromosome, and they're called haploid. Now, thank you for your time. This concludes all I have to talk about. I ask that you have an amazing day. God bless.